Thanks, Dan, for the very kind introduction. But that's true that I'm, jack of all trades and master of none. So if you tend to ask too deep questions, I may not know the answer because there are many things I have to know. So yeah, as Dan said, I have been, doing a lot of GWASs in my life. I think, many hundreds of GWASs, and led also some of them. So I know a little bit what I'm talking about. So, but more recently, I'm involved in personalized medicine and using the genetics in the healthcare. So if we have time, I will show a couple of slides in this topic as well. So you already have gotten a quite very good content on the biology and the structure of the genome. But what I wanted to talk about, how we can use the GWASs or association analysis to a little bit learn more about the biology, and also discuss a little bit the limitations on how deep and how easy it is to do those analyses. So this is an intro slide, so it's just a random plot from the genome-wide association study. It's called Manhattan plot. Sometimes they're also like, I don't know, Dubai plots or something like that, because the association peaks are just so high. And if it's like gender stratified, it's like a Chicago plot, because you have both the skyscrapers and inflection or reflections on the lake. Anyway, so it's a usual one. I think it's from the EA3. Looks like EA2, where you have your associations. These days it's like in high hundreds or even thousands. And you can do all kind of interesting things that you have been discussed for the past 2 weeks. But if we take any of those association regions in the genomes and we zoom in, and this is what it's called a locus zoom plot, where it's basically the same as this, that here you have the the variance, and here in the y-axis you have the statistical significance, on x-axis the physical location, and there is also shown the LD between the markers. And here are shown all the genes that fall in this region. And as you can see, there are like 30 plus genes in a window of what is it, like 800 megabases, or 800,000 base pairs. So basically to question which of those genes in this region, or any of those regions, is actually linked to the disease or is having any effect on the phenotype we are studying, is actually quite complicated, because there is no further insights from this association peak than as it is. So any of those genes could be the causal gene. But it also gets worse, because we also don't know which may be the causal variant, because we may pick the one that has the lowest p-value, but it doesn't mean that it's actually the causal variant, because I will touch this on the next stage, because some of the information may be missing. There is like other problems, and we also don't know, even if we figure out the variant or the gene, we don't know which of the tissues this is important. Does it affect in our big toe, or in our heart, or in our brain, or a certain region of the brain? Such a flat plot doesn't help us further. And so I'm going to talk about different ways how you could try to figure out which is the causal variant, which is the causal gene, which is the causal tissue, and what is the biological pathway all these results are hinting towards. So the first important thing to understand the limitations of a GWAS. So you already got some slides about the technology, how the genome is profiled, and most often what we do, we use those genotyping arrays. But it's important to remember that the genotyping array is just a proxy of a genome. It's just used to describe the structure of the genome. And here, quite simple cartoon, let's say a random section of the genome for people, and there are the letters in gray are the ones that are the same between any individual, any humans, and then there are certain places where there is a difference. Those are called single nucleotide polymorphisms. They happen like, once per 300 base pairs, and you will get such a picture if you do the sequencing, so the full genome or full exome sequencing, where you read every base pair. But most of this information is actually redundant, because what is gray is the same between all the humans. But even if I, for example, take my genome and Kaili's genome, who is also happens to be Estonian, then we have on average like 3 or 4 million differences in their genomes. But if I pick anyone like more exotic, then actually the number is can be much higher. So actually, but doing sequencing the 3.3 billion base pairs is very expensive, and it doesn't make sense because there are lots of redundant information. So what we can do, we can like sequence a bunch of people and then select only locations in the genomes that are or have a variation within it. But even if you like look closer, there are also structural correlation, LD, between those positions that vary. And so actually we don't, and this is redundant information, so we don't need to type all those variable positions in the genome, but it's enough if we use some computational algorithms and pick the positions in the genome that describe or capture most of the variation, or we can use those 3 SNPs to actually decide which of the 4 haplotypes given person is carrying. And those, what is called tag SNPs, those are actually what ends up on the array. And in the modern arrays, it's the number of variants is something between 700 and 2 million positions, and this using such array costs you something between 20 to 40 euros or dollars per sample, depends on the design. So it's very cost efficient. You can do tens of or hundreds of thousands of people in your study because you can afford it. And then what happens if you have a reference panel, like the 1000 genomes reference panel or haplotype reference consortium reference panel? I hope those have been talked about. Then you can actually, just knowing those correlations, you can actually impute or infer all this information up there. Although there is like probabilities, you can get the information back. And so for that reason, as on the array, we don't have selected variants based on their causality or based on their biological function or based on their prior knowledge about the disease associations, but just because they're very good proxies or like beacons to deconstruct the haplotype structure, they're selected on the array. So anything on the array that gives you a nice association doesn't mean that it's anyway, being causal or anyway even close to the real causal location. Is this clear? Because this is like, it's the core concept why interpretation of your raw results from the GWAS is not very, it's a little bit tricky. And so right now we know that, what are the tricks we can actually try to find, the causal variants. So something that is very, very simple, and this is an example from the height paper Jonathan mentioned yesterday. So there I think had like 200,000 participants, and we found at the time quite large number, like 700 independent hits. And then we used some basically took into account the LD structure of different variants, and basically what we did, selected the most significant variant from the genome from this region, put this SNP into the model, repeated the association analysis, so until there was nothing significant left anymore in the data. And then we actually found out that there are, in one region, in the size of, again, like around one megabase, there are, 6 independent haplotypes or associations. And now if you look into those those independent variants in the locus, you can see, and you look the genes that fall within it. This ACON gene is, for example, very important in cartilage development. And for example, the actor Danny DeVito, is very short, and he's short because his body is okay, but his legs are very short. And he's actually this gene is not functional in her genome, and that's why the bones couldn't grow, because the cartilage is very stiff. But there are like a couple of other genes in the region that have been linked to Mendelian form. So one way how to pin down a causal variant in your locus is like look if any of those genes are linked to any of the like Mendelian or birth syndromes. The other one is like look for doing this conditional analysis, seeing what's more there significant. But what you can do, you can see if those variants that show independence, is there in strong LD any coding variants? Because again, like in the imputation, because we have probabilities, some variants impute better than others. Not even here, the most significant doesn't mean that it's the causal. But if there is, let's say, non-coding variants in strong correlation with your SNP, and non-coding meaning, or coding variant meaning that it's contributes in the protein sequence, and if it's like missense or that it somehow changes the protein sequence, then the probability that, or the chance that this is the causal gene or causal variant is much higher.
So I just want to add that one of the things you mentioned, that looking at the most significant SNP and then controlling for that and rerunning the regression, that's the COJO method that ISU taught.
Yeah. But this is basically visualizing what doing the COJO and put this into context, and then looking those independent variants coming out and seeing if there are any missense variants or something that changed the protein sequence. Yep.
So just to be clear, it doesn't have to be the coding variant, does it? Because
No. Yeah. This is will be like 20 other slides. Yep.
Without making protein, but it's in some sense it's empirically been found that if it's a coding, if it does coding, it's more likely to be a powerful SNP.
Yep. But in I would say 98% of all your associations don't fall in genes, or not even close to the genes. So those are very rare cases where your associations falls within the gene, and this gene has been implicated in Mendelian this syndrome analysis that this is a causal gene for some kind of early onset disease. But yeah, of course, many of the variants are even not close to the gene, so then it gets much more complicated. And then there is following up on Mali's question, there was a recent paper from Giant, again looking into height, and there what we did, we had a little bit different array design where we had more of those very rare variants and very rare coding variants on the arrays, and there we also had a massive sample size, not yet millions, but still. And here it's are all the the independent best associations, and here you have the allele frequency, so it's from very rare to ultra common, and here you have the effect sizes, and I think the things that I think all those are coding variants, I guess. But anyway, so you can see that if the frequency in the population is very low, then the chance that, or then there's a correlation that usually that means that the phenotypic effect is also much larger than for common variants. For example, I think this this variant, just one single SNP explained or had an effect on, I think, almost half a centimeter, which is huge. But at the same time, it's ultra rare, so it doesn't actually explain a lot of the population variation or missing heritability. But it's important to know that's the correlation, that the rarer your association variant, and the impact of the phenotype is usually much larger. But and another very interesting study from Andrea Ganna, where we contributed our genomes, they looked for all this coding missense variants in the genomes, and then they looked and classified those according to the where are the genes expressed. Are they brain expressed or not brain expressed? And I think the and there the genes that are in brain expressed had much higher impact. I think the locus was again on some neurodegenerative diseases. And another thing, when we looked at how much of the phenotypic variance gets explained, either looking at those, for example, pathogenic CNVs, we didn't talk about those, but basically where you have like missing some genes or have some genes multiple copies. Then when you have like excessive recessive variation in your genome, then you have your this disruptive or damaging, very ultra rare variants, and then you have polygenic risk score. So here, the polygenic risk score still explains far more of the phenotypic variance, but all these times, this disruptive rare mutations actually help you to figure out what could be the causal gene. So now we go into the so this was only the 2% of your usual associations, because those fall into the genes, but most of your stuff is outside the genes, like all the different kind of regulators. And so this is just another cartoon to demonstrate how many different regulatory mechanisms there can be to every gene. So the gene will be the coding variations, the exons and introns that where you get the protein sequence will follow there. But there are many regulatory regions up to like tens or hundreds of thousands of base pairs upstream from the gene or downstream from the gene. And quite often all your genome-wide association signals fall into those different regulatory regions close to the in the proximity of your gene. And what does a regulatory sequence means? That is like all your the different transcription factors or regulators, modulators, enhancers will have some kind of sequence that they bind in the genome. So this is just one of the example of often a signature for a random transcription factor, and you can see that when there is just one letter here, it means that there cannot be any variation, because if there is like a different letter here, instead it's C instead of T, the transcription factor is not binding at all. But if the size of the letter, there are several letters, then it means that the binding affinity of the transcription factor just changes a little bit. So, and this way, for example, here it's like it's not A but it's C. It means we can like make approximation that the transcription factor binding affinity is like 3 times lower, which means that we get 3 times less protein, and that's why maybe in the end our blood pressure is 1 point lower than others. And why this? Why most of the genome-wide association signals fall into the regulatory regions is because there is much more plasticity of the genome and much more plasticity towards the environment. Because if you are switching off or on a gene completely, it's completely different. It's not very plastic. But if you can just tune, like how what's the path of your song, like then it's like has much more variability. It's like driving a car when you are missing the wheel, or you just have the pressure in the tire a little bit different. The driving properties is completely different, and I think it's the same holds here in the in the genome. And as I mentioned before, the gene expression gene expression is a very nice proxy to disentangle the causal variants. And there is an excellent study, it's like GTEx, where they have, I think now already 1,000 people, where they have up to 200 tissues, gene expression profiled, and the genetic profiles as well, so they can do a lot of EQTL mapping. But they can, in general, also look at the gene expression levels in different tissues. So something is highly expressed in brain, and something is highly expressed in liver. And usually if a gene is highly expressed in brain, and you're looking into the phenotypes related to brain, then probably this is the causal gene. So yeah, I encourage to check any of your association variants that you get from the GTEx database, because there you get a very nice overview, where it's expressed, and does your variant have any effect on the gene of interest. But I know Andre already showed you a couple of slides about this concept of mapping QTLs, or looking like.
Do you have a question?
Yeah, I have a clarifying question.
Yep.
So when we have this sequence,
Yep.
here, this is kind of tell me if I'm wrong. I'm just trying to get a sense. There's 4 possible letters in some places.
Yep.
It's like a theoretical idea, like if it's a C, you would expect defining very strong versus G, but in reality, you only 2, or
Yeah, in principle, yeah, it's mostly like the most of the SNPs are actually selected to be biallelic. So in the population, you always only have 2 alleles, but there are many SNPs actually which have 4 alleles. But it's like very complicated to put those into array, because Andrea explained it's usually like two colors used. So that's why you only pick the two allele variants, because then it's like very accurately you can type it.
So this is not theoretical.
I think the question is, how do we know the binding affinities for all the possible things?
So mostly they are predictions, but sometimes also people have designed all kind of different versions of this binding motif, and then did like verifying experiments, like what's the how strongly the transcription factor binds with certain sequences. So it's partially predictions, but some of those things are also experimentally validated.
I just want to add to that, because so I think some of this is like there's 2 types of experiments, so the SELEX experiment and ChIP-seq, where basically they look at enrichment of the binding partners, the DNA binding partners. And so those basically those frequencies that you look at are kind of often the consensus sequences of TFs binding to that. You can either do it by randomizing the sequence and then look at the enrichment of the binding partner, or the ChIP-seq experiment.
Yeah, so basically you take this specific transcription factor, bind it to the genome, then dissect everything else where the transcription factor is not binding, then sequence everything, and indeed then you just look what's the consensus, like how many cases you have, like a certain letter here or there, and then usually where there are like those, usually only 1 or 2 letters, then this is like has to be there, and everything else is kind of random. But it still, may have some effect on the binding affinity. Okay. And so one way how to then, find those, if, for example, your genome-wide association region is indicating for some kind of binding sequence, is actually doing, expression QTL mapping. And, what's the, the point, what's the reasoning behind it is like, for example, you have a variant that falls into, promoter region or some transcription factor binding region, then having a different genotype, the gene will have a different expression levels, and this is demonstrated here that with different different genotypes, you have different, a linear correlation between your genotype and your expression levels. So basically running, if you do a EQTL mapping, you're basically running like a local genome-wide association study or association study. Usually it's done like plus minus 1 megabase from the gene prescription start site, and then you just skim this region and see which of the positions or variations has this kind of linear relations, and then this has indications that there may be some kind of regulatory variant there. But you can also think are much more complex, so this is called usually cis or local regulation, but you can also get more complicated if it's trans or distant regulation, and this is even even between chromosomes. So usually the LD is only valid within a chromosome, but those kind of regulatory networks can expand over chromosomes, and here could be, let's say, chromosome 2, you have your SNP in transcription factor that changes the transcription factor configuration a little bit, so its binding affinity changes, and then this transcription factor can go to, let's say, chromosome 15, and then based on the genotype in this in the gene X, the expression level of gene Y will be different. But those effects are much weaker, and you need much larger sample sizes to find this kind of trans effects or distant regulatory effects. Yep.
Speaking of sample size, what are the typical sample sizes?
So for any type of this kind of endophenotype is a phenotype that you can read from the from within the biological mechanism, like expression or protein levels or metabolomic levels. They are actually you can already get signals with a couple of hundred samples, but then you have to have for example, sorted specific cells from blood, or get skin cells. Then it's 200 is you already may see something. But if you're working with, let's say, blood, which is I don't know, many tens of different tissues actually, or cell types, then you need thousands. So this study I'm showing here is 5,000, and we are just hopefully soon in press in Nature, where we have 35,000 samples. And there we find basically this type of cis regulation for almost all the 20,000 genes. And we demonstrate that there is a lot of trans stuff as well. Yep.
This information, is that similar? Do you ask some statistics?
Yeah. So it's on my second to last slide. There are links where you can get to this data. So yeah, as I said, we did already quite many years ago the first study on blood EQTLs, and looking for and the aim was indeed to help with finding the causal variants in the GWASs, and this data has been used I think in more than 100 GWAS papers. And so what we did, we did this local mapping, and then at the time with HapMap2 panel, we found that 20% of all SNPs that you have some kind of EQTL effect, which means that if you just look up your best SNP, and if it has EQTL effect, you think you have found the causal one, it's not actually accurate. You have to look the full local association plot for given gene to decide if your variant is, your variant may have 10 to the power of minus 47, you think it's awesome, but the best variant may have 10 to the power of minus 370. So it's always you have to put your variant into the context. And we also saw that almost half of the genes expressed in blood, there was one EQTL on it. And in this graph, it's also here you have the distance from the gene. So it's up to 250 kilobases what we looked for, and this is the effect of the variant on the gene expression. And obviously, the closer the the regulatory variant to the transcription start site, the stronger the effect. But there are even 250K away, there are variants that have a very strong effect on the gene expression. So it's like all over the place. And here the same is, you can see the distribution or density plot for where are your causal regulatory variants from the genes, and usually it's very close. So that the one of the approach to find the causal gene, you take the closest to the of your association variant. It's kind of accurate, but not always. And we can skip those ones. And then, as I said, we also looked into the trans effects, and as this is computationally super heavy because you have to run basically 20,000 GWASs, and then you have to get it from 10 cohorts, and then you have to combine those results. It's like awesome, I don't know. One file that we uploaded is like more than 2 terabytes, so it gets like really awesome. So we didn't do it. So we only focused on the variants that have been associated with GWAS before. And then we found at the time with 5,000 samples that 8% we tested like 5,000 GWAS catalog SNPs, and of those 8% had this kind of distant effects. And this is what it's called a circus plot. Here you have the chromosomes in the circle, and here you have the genes, and in the interim you have the SNPs, and you can see that there are some hotspots in the genome that affect many genes, and there are some that don't. So it's quite impressive. 95% were like interchromosomal correlations. But as I said, we 5 years ago we had the study with 5,000 samples. We found that there are 8% of SNPs in the GWAS catalog have this kind of trans systems effect. Then we repeated the analysis with my postdoc, and with 14,000 samples we found that approximately 20% of all GWAS catalog SNPs, and we tested 10,000 SNPs at the time, had this kind of systems effect. And then current version that is in review in Nature, I think it's even higher. I think it's 41% of all GWAS catalog SNPs demonstrate some of this long distance regulatory effects. And this, why I show this is it is like to demonstrate that it's not as easy to just take the closest gene to your most significant SNP in the locus. It can be that it's just a gene in a completely other chromosome because of those system regulatory effects. But not to have it like too theoretical, one example is what we had in the Wester et al. Paper. Basically we took a very strong SNP known to be associated with prostate cancer, and then we looked locally, which genes this variant affects. And then, we got 4 genes, and then we looked, genome-wide, which gene expression was affected, and there we got, I think, like more than 10 genes. And then what we did, we used some tools to predict the function for those genes. And in short, if you look at the genes that are in close, they're not very much related to cancer biology. But if you look those this distant regulation genes across 10 chromosomes, then those are already much closer to the cancer biology, like the angiogenesis, like the regulator or factors that help the blood vessels to grow into the tumor tissue, or DNA repair stuff, so that the cancer can go wild because there is no regulation on it. So this is how you can just having one SNP from your genome association study, you can actually infer quite a lot of about the biology of this of the phenotype of study. And you can at some point the colors are not great, but basically here are both the cis and trans for given SNPs, and then you start to have this kind of aggregations where you have like many SNPs and many genes come together, and you can infer pathways or biological functions from here. So any questions for the EQTL part? Yes?
Well,
so in this example, there's one SNP with so much effect. But on the other hand, like the whole idea of the polygenic score is combining a large number of SNPs with minimal effect. Like, how do you reconcile these approaches and when to choose which?
So there is no conflict between those things. It's just like one of the examples that one gene can affect so many genes, and that's actually I think the power of the polygenic risk score and the deep polygenic risk score. So you don't get just the genome-wide significant SNPs, but you get like much deeper ones, because then you capture most of the biology, and you capture because it's not just one biological pathway that leads to cancer, for example. There are like tens and tens different pathways that contribute to the risk. And so if you take your polygenic risk score and you have it very deep, your score captures almost all the disease biology and almost all the risk or effect on a phenotype. So there is no so there are completely different problems is to like finding the causal gene and then trying to design a drug against it, and then using the polygenic risk scores in understanding the general processes in a body or disease risk, or use in healthcare. Yep.
What's the biology of these long-range trans effects, like between chromosomes? Does it have to produce some RNA to go with the chromosome?
So I think there are like 4 different ways. And one is, like as I showed, like this, your SNP affects either the expression level of a transcription factor on chromosome, one chromosome, or it falls within a transcription factor, and this way changes the configuration of the transcription factor. And then this transcription factor either having a lower concentration or changed binding affinity goes to other locations in the genome, and there has an effect on the different expression level of your downstream genes. So this is one effect. The other one can be that your variant falls within a non-translated RNAs or not. Yeah, so it's basically like this non-coding RNAs that can then go and bind with your mRNA and just get it degraded because there are some mechanism for that. So this way you can have the distant effect. It can be that you have we like to think, or you like to think, that the genome is like nicely in the chromosomes, nicely rolled up, but actually in the cell it's like all kind of like a mess. It's real. So it's not that chromosome 1 is next to chromosome 2. No, it can be like it's a complete mess there. And so there it can be that your different regions of the genomes just get very close to each other, so there is some kind of binding or interaction between those regions. So this way you have the distant effects. Or I would say the also a possibility is that your SNP affects some kind of endophenotype. For example, it has a huge effect on your cholesterol levels, and then the change of the cholesterol levels in your organism changes the gene expression of many other genes. So, but this requires quite a lot of biological experiments to work out what is the mechanism. So we have looked on enrichment, and you see enrichment on the transcription factors. You see enrichment on the non-coded RNAs. You see enrichment on this kind of chromosomal binding, and we also have examples where you have this endophenotype is being the cause. So it's quite a messy thing. And that's my point, to show that understanding the biology can be quite tricky. So finding the right tissue, as I said, like one way is like going to the GTEx database and see, if you know what is the causal variant, you can see, what genes are affected by your causal variant, and then you can check in which tissues these genes are expressed, and then okay, yeah, this is like brain or liver or the big toe or whatever. But usually in a genome-wide association study, when you have hundreds or even thousands of loci, this kind of picking one by one is actually not very clever idea, because you would need like a like twice or thrice as many people as in the room to look all those genes. So this is used to be in the early days of GWAS, where everyone read all the papers in the PubMed about the gene and then wrote the paper. But now it's not even possible. So I was part of. Oh, probably it is possible. James did it. James did it for EA2 and also EA3. So it's crazy. But James knew all the the textbooks also by heart. So it's anyway. But in principle, you have a lot of loci, and you have to make some kind of infer some kind of biology from there. And this is a tool, still very much of use. It's called Depict. There is also a link in one of the last slides. But basically, what this tool does, it takes your genome-wide association results, then defines, the independent regions, and then from those regions, he picks all the genes that fall within this region. This is just like how you define a region and the cutoffs. It's a lot of modeling, but basically you take all the genes across all your loci, and then based on those, all the genes across all the loci, you can start to ask questions. Which of the biological pathways are enriched across all your regions, in which tissues this kind of enriched pathways are present. So you can do this kind of prediction-based stuff on you can prioritize the pathways. And now when you have prioritized the pathways, then it's also you can go it's a little bit circular, but then you can use those pathways that are enriched and pick out the genes that actually fall in or are active in those pathways that have come from the first place. So you can first get the pathways and then use the pathways to prioritize the genes in your loci. And you can do exactly the same way that you can, if you have your pathways, and then you can look the genes falling in the particular pathways in which tissues they are mostly expressed in. And this way, actually using all the data, using all the information, you can make predictions quite efficiently on the causal genes, on the causal pathways, and the causal tissues. And why this tool was kind of revolutionary at the time is that it's actually goes beyond the published literature. Because how is it constructed? It actually the team downloaded all the gene expression profiles available at the time. It was like 80,000 or almost 100,000 gene expression profiles. And then they constructed a lot of networks and co-expression networks. And basically what they could do is like if, let's say, 50 genes annotated to be involved in cell death, and there was like 51st gene having exactly the same profile, they could predict that this is probably also involved in cell death. Or opposite, that if there was one annotated to be involved in cell death, but it had completely different profile, that it's probably like wrongly annotated. And this way, actually, this tool really, I think, was revolutionary in GWAS, because whatever disease or phenotype you put in, you get out the relevant biology. For education, we got brain stuff. For height, we got the bone stuff. You put in diabetes, you got liver and pancreas. It was really, really, really cool. But it only works if you have many loci to work with.
What's it called? What's the tool called?
Depict. Yeah, I think I said it, but it's in the link is in the end as well. And this is just one of the examples. I think it's from the EA2 paper, and there is the similar example from EA3. I just didn't update my slides, but basically we had 75 loci, and then if you take all the genes across all those 75 loci and then ask, in which tissues those genes are most expressed in. So then this is the graph based on the GTEx. There you have like all the different tissues, and there is brain, different regions of brain, and basically you get only enrichment for the educational attainment loci for brain regions. And this is another one where we looked into the expression levels of the genes in the loci in a developing brain. And there again, the genes predicted to be involved in the educational attainment. There is a higher expression in general than the background genes, but then we also saw some interesting mechanism that there was like higher expression in the prenatally than postnatally, but this signal I think disappeared in the EA3, was it? Kind of.
It doesn't disappear. It just means that the first few genes, it looks like that, more prenatal, and then the next 1,000 is black.
Yeah. So basically, it means that there are like all the different development phases are important in the brain. First, it's like how the brain is structured, and then how the signaling happens, or how the synapses are pruned. Otherwise, you get like overstimulation and stuff. So it's again like using the different databases, you can infer quite interesting biology. Yep.
Is that the tool also depict for this temporal expression? How does one find?
Yeah, this is I think there is no tool for that, so it's like a custom-made analysis by that. But the temporal expression data is possible to download, and then you can. But James probably can explain more.
So, you go to the Allen Brain Atlas, and you can download it. Also, I believe you can download it from Tuna's website.
Yeah, from the
There's one
depict.
that's GitHub. Search that, and you should be able to find somewhat pre-processed already the brain span data. It comes from it's tied to a paper from Nature. It's old now, 2001. But there seem to be a few data sets like this. It's still extremely valuable.
This is a collaborative project. Brain span is it's available from I can send you the link also, but they have multiple developmental periods. They actually span 16 brain regions.
Yep.
Related questions. How fast are the annotations improving, and are there still what areas are the annotations really still of visible property?
I think it's improving slowly but steadily, but I think still like 25% of genes no one knows what they're doing in the system. And I think probably the brain stuff is the most annotated because there is a lot of work there. And there's also a lot of brain genes anyway. So it's mostly those annotations, require animal work and cellular work and lots of pathways and lots of to understand what's going on. And even then, you may not know all the functions because you annotate only some of them. So that's why the prediction framework was particularly interesting, that we could predict many biological functions that are actually not relevant. That made the publishing of the results tricky, because we said like those genes are predicted to be involved somewhere, but actually there is no published evidence on that. Yeah. And this is just like a very quick example of how you can, using the GWAS, understand, the biological mechanism. So we had 1,000 Estonians sequenced, low and measurable number, but we had blood cell counts available, and as we didn't have any missing information in the genome, we had quite good power. And so with 1,000 people, we found 19 signals across 10 phenotypes. So pretty good. And the strongest one, but the bad thing was that everything was already published. So the strongest association that was a regulatory variant was novel and enriched in the Estonian population. So here you can see the Manhattan plot. So basically it was flat, and then there was 2 association peaks. And then we replicated the signal within the cohort using electronic health records. And then when we looked into as I said, it's a regulatory variant. And then we looked in which tissue type this regulatory region is active. Now, this chip cheek, or was when you kind of try to figure out if the genome is open, if it's euchromatome or where it's packed together, then here we can see that there is for this, we had the phenotype of basophils, and so the progenitor cells for basophils, actually this was the only place where this regulatory region was open, and there was another association as well, and there this regulatory variant has a much broader binding affinity, or had like much broader regions open. And then what we did, actually we don't have slides, but what we did, we actually used CRISPR, so gene editing or genome editing experiment, where we actually cut out this regulatory variant in the cells, and then we were able to demonstrate that actually the differentiation in the basophils didn't work very well. The basophils were kind of misforming, and so this way demonstrating that this regulatory region is like crucial in the biology of the basophils. But this is just one example how we can use not just gene expression, but also epigenetics, or this chromatin structure on one locus and one phenotype. But this is again an example from the EA2 paper, where we did a similar experiment again across all the loci, and asking in which of our genes or where are the gene expressed or where the genes are active in which tissues across all the loci that we had. And while the data is very, very messy, the brain still showed like the strongest signals. The blue is educational attainment, and the others are anthropometric traits as a control. So here you can see that the brain, the education has the strongest effect. Whereas like as a positive control, here we have height, we have bone, and it's the bones, the height genes are the most active. So this is another way how to finding out which of the tissues which of the tissues are important for your phenotype, like looking at this genome-wide effect of chromatin structure.
So Tõnu, just want to point out that the method you're showing here, the statistical results are from partitioned LD score regression, which Raymond talked about.
Yeah, I see that like there are so I'm not even trying to explain the mathematics here, but one thing is like to see the mathematical algorithm, and what we can kind of theoretically do with it. And this is like maybe this was also demonstrated, but this is like example how you can use this LD score like partitioning to actually understanding the biology behind it. And there are, I think now even probably much more updated methods and much more nicer figures and examples. But this is kind of like one of the early examples how you can use the chromatin structure to understand the biology. And then also like going to the genes, I already showed this enrichment methods where you can use all your loci to figure out which could be the causal genes. But another trick that has been used is actually to look into the Mendelian genetics or into the genes that are causing some different growth or like different birth syndromes, and then because like if you have, it's the same analog, like if you have a gene missing or not functional, then you may have a major phenotype, for example a big effect on brain development or cognitive decline or something like that. Whereas if you have the regulatory mechanism tweaked a little bit for given gene, you may have just a little bit, you will maybe a little bit smarter or a little bit slower in thinking, but other than that is cool. So this is again example from the EA2 where we looked if the loci that came up from the association analysis, like are they enriched or are there genes that are involved or are linked to, for example, Mendelian forms of schizophrenia or autism or intellectual disability, and then we see actually a quite large, overlap, and this provides the confidence that we are finding the right stuff, and their statistics are correct. And this is just to demonstrate that what we did, we used tried to look into the enrichment, and if we took, for example, there are like the different intellectual disability gene, autism gene, schizophrenia, and then we also had as a control the growth genes. Then you can see that there is a substantial enrichment for intellectual disability, autism, and schizophrenia, but in growth related genes there is like no enrichment for educational attainment, but the big one again for height, which is like a negative control. So one thing is look for this Mendelian stuff. Other one is not used anymore, but it is that you can look into literature. So there were tools used where you put in your SNPs, and then the tool looked across all the PubMed abstracts. Are there gene co-mentioned in abstract together with the given SNP, and this way you could also prioritize your genes. And this is example from the height study where with just 200 loci you can prioritize some genes, but if you have a substantial more loci, you can use the literature to prioritize more genes. But this tool is not very much used anymore because there are so much more knowledge that you cannot really use, because the GWAS is kind of biasing your literature mining tools. Okay, and finding the pathways, the same way as I described, you can like look across all the genes and loci, and then start to construct those different pathways. And this is again from the EA2 paper, but and you can see that the pathways that came up from this enrichment tools, and this was again from the PICT that we used. There are many words. James can talk hours about those, but basically it's like it's like self-explanatory that if you look the pathways that come up, that actually it's something to do with the brain that we are studying. So there's also the many times the question that if you have a very weak signal from the GWAS, very often your polygenic risk score explains couple of % of the phenotypic variation, and if you take a single SNP, it doesn't explain almost anything. In height, it's like the strongest explains like 1/3 of the millimeter, I think, is the strongest set. So many don't explain almost anything. So can this be used to learn anything meaningful about biology? And this is it was not very recent, but a very nice paper. It's the schizophrenia, the first, this massive scale one where they identified more than 100 loci for schizophrenia. And from there they, found out, or it's well known, that there is the, HLA region in chromosome 6 has a huge impact. Like this is the peak here, I think. A huge effect on the schizophrenia development, but the MHC region is it's very complicated and very complicated regions, and there are different compounds that contribute to the disease. And then there was a accompanying paper where they took this insights from the GWAS, and then they figured out all the different haplotype structures within the very complex HLA regions, and then they tested all the different haplotype structures in cellular models, and then they found out that a certain configuration or certain component in there, it's called complement component 4, is causal, and they were able to demonstrate that if this complement is overactive, then actually too many synapses get pruned for your brain cells, and this means that there is not enough connectivity, and this way may actually then your brain functions normally because there is no overstimulation. Whereas if this complement is not really working, the synapses are not pruned, then you get like overwiring, and that's why schizophrenia happens, because you have more friends than you actually have. This means that there is like this more information that you can handle. So, but at the same time, you can basically, and now you can, if you're understanding that, you can design a drug against this biological mechanism, either for the specific complement component 4 or going for the pathway. And with this, one insight, you may end up with a highly successful drug that may, hugely benefit, the patients. So my point here is that, that the SNP has a very minor effect on the phenotype or the risk of the phenotype. Doesn't mean that once you understand the biology, that the drug wouldn't have effect. So, for example, the similar example is statins. The drug used to lower cholesterol. There, the SNP explains only, I think there it was almost a % of cholesterol variation. And then there was a drug designed against this protein, and then the statins lower your blood cholesterol levels at least 30%, while the SNP itself only explained 1% of the variance, or maybe 2%. So that's the example that the weak statistical signal doesn't mean that the biology is weak. So those are the links. Should be active. If not, I can update those. And then I think I have like 5 minutes. I just very briefly wanted to show 2 slides about the personalized medicine stuff that we do in Estonia. So basically, why we are obsessed with genetics is that genetics is there on day one, and it's different between us all. So there. Good news is that no one has all the bad variation in the genome, but bad news is that everyone has something wrong. So this is, everyone has something that is not working. So live with it. But it's important to know what is it wrong, because if you think our risk toleration of basically the disease risk and how much we tolerate the bad lifestyle. It depends on how much bad genes we have, because if you get older, the system gets not functional anymore, and we add bad environment, smoking in primary school, not secondary school probably, drinking beer in high school, and quitting physical activity in university, may get to your picture of risk tolerance kind of full, and in the early 40s you get the blood disease. So that's why our incentive is that if you know the risk, genetic risk, we can either motivate people to live healthier, or we can introduce some kind of interventions to kind of get this lifestyle risk or physiological risk lower. And I'm just showing you one graph. So just focus for this. The screen normal distribution plot. So this is the distribution of the polygenic risk scores in a big population. So it's normally distributed. And as in the normal distribution, there are only very few people who are either have a very high load of the bad alleles or have very low load of the bad alleles. And just to simplify, let's take breast cancer, and let's say there are 1,000 loci associated with breast cancer. Then actually the person can have either 2,000 bad alleles because they get 1,000 from mother and 1,000 from father, or they can have 0 because they're lucky they get nothing. But those are very few people. And while I think you mostly talked about the predictive ability and how much variation gets explained and so on and so forth, actually in medicine, that may not be the most relevant thing to predict the absolute risk for everyone for a given disease. But actually, what is much more meaningful is actually finding the fraction of finding the people who would have the highest risk and would most benefit from the intervention. And to illustrate this concept, so this is a Kaplan-Meier curve for breast cancer. So what we have here is like the age on the x-axis, and in the biobankers we have like people all over the age distribution, and we have followed up them for 20 years. We know what happened with them, and here we have the cumulative incidence of breast cancer in a given cohort at a given age. And here, you know, 3 lines are important. So the red one, this is the top 3% of this distribution here, and here we have the dotted line. This is the population average without any genetics considering, and there the dark blue is actually the bottom 25 distribution. So the folks like here. And now if you follow them through the age, you can see that by age of 70 or 75, 15% of the people with the highest polygenic risk score have developed breast cancer, or actually women. But at the same time, the population average is like, 7, and for the bottom risk groups, it's like around 3. So we see that by age 75, there is something about like 5 to, and some diseases it's 10 times difference in the disease prevalence. And if now we think in the context of what the state is doing, usually, they're offering mammography screening programs for females, but it only happens in the age, between 50 and 65. And now what if we take the age 50 and we look at the population average there, the prevalence is 3%, so it's, makes complete sense to start at this age, because, the prevalence isn't very high, but for this very low distribution of the polygenic risk score, it's even like below 1%, but for those unfortunate ladies who have this very high distribution, among them, it's already, more than 5. And it's actually very much justified to start the screening for those in the top 5% already in the early 30s, and go, until, their full lifespan, because they are very much, higher risk, because they have at, on 1% already, between 40 compared to the lower risk group. So this is what we are currently doing. So the 50,000 participants which we have, there 1,000 females have been selected from this high distribution, and then they are invited back to see the oncology regardless of their age or family history, and same thing goes on for cardiovascular disease, and probably we'll launch the glaucoma program as well. So this is all I wanted to say, and happy to take maybe a couple of questions, and then we have a break.
I have a very general question about functional genomics and social science. I understand the importance of functional genomics, the things you showed about validating polygenic scores or G1 studies, and also for the biology. Is anybody using that information in social science research, and how can we do it? What is that?
So the question is like, the functional, the functional genomics. I think there was Lauren asked this question yesterday. Like, can we, for example, start to construct our polygenic risk score based on the underlying biology? And probably we can, because we can figure out for each loci what is the kind of like the probable gene, causal gene, and then we can annotate this with the pathway, and then we can try to do pathway-specific polygenic risk scores. And I think we try to do in boldness, because in the boldness genetics, it's much easier. It's like, I think, 150 loci in total, explain quite a lot of the variance, and so then we try to do it. And we got some insights, and I think this kind of things could do also for the education attainment or the risk behavior or any of the personality traits. But what is there necessary? We have to have probably even larger numbers of loci, so we could actually, dissect the score, because like every time we, kind of leave out a bunch of loci, actually our score predictive ability, gets lower. And so if you at some point, if you dissect it, across 20,000 pathways, you don't have any power to ask any questions. So that's also this is one point. Another point is like in social science, how important is to know the causal gene versus like having an instrument to control your experiments for. That's what genetics, this score you can use. If you don't have, parental education, but you still want to have a proxy for socioeconomic status or something like that, you can start to use the genetics because it works the same way. So, but I don't know if your experiments gets any much more valuable for social science if you start to dig into like very much on the gene expression or something like that. I think the genetics is already doing a very good job. I don't know who was that before.
I just want to add, I think for Mendelian randomization studies, they're most convincing if you can argue for the exclusion restriction on grounds of understanding the biology. That's not currently the convention, but I think it should be.
Yeah. So that's the right point. Like you cannot start to do Mendelian randomization if you don't understand the biology, because there are all those things you have to meet that it's independent and this is the right the mediation pathway. So that's really the point. That's why the smoking stuff works. Is that it's
an example yesterday Mr. Big for smoking and then the two alcohol flushing reaction.
Yeah. So the biology understanding is extremely important for the Mendelian randomization. I think Lauren.
Yeah, I just wanted to mention too that a colleague of mine at Wisconsin, who's in the Department of Biostats, does have a paper that has developed a method called ANOPRED that constructs polygenic scores based on functional annotation. And so it uses the Bayesian method to kind of shrink the betas based on kind of what types of functional annotation you have.
Yep.
So I'm using that in some projects, and I think it's a promising thing.
But it's also like a little bit what is the question, like what type of question you try to answer by, kind of dissecting your polygenic risk scores.
So in my case, I think it's quite fascinating that the majority of citizen loss lag cancer regions, and I think those could also be regions that are more susceptible to the environment. And so perhaps we could get more out of gene-environment interactions with a stronger signal if we construct polygenic scores that are kind of capturing aspects that are more sensitive to the environment.
Yep. Yeah. But in general, as I saw, like anyway, most of them are anyway enhancers or regulators. So, but it requires, very large number of loci in order to, when leaving stuff out, you still, keep the predictive ability of your score.
So for Mendelian randomization, ideally you'd be saying not just what biological mechanisms there are, but what biological mechanisms there couldn't be. So how good could you do at saying it's really very unlikely that there's thus and such biological mechanism, and therefore I can exclude it in the Mendelian randomization?
One way how to answer your question is that there are big genome sequence databases, like one of the most known of them is the GNOMAD, and there are developed statistical methods to say how unlikely it is that the given gene would carry a mutation without any phenotype, like basically the intolerance of mutations. And then based off this information, like if you have a sick child, you do the sequencing, and this gene that is highly unlikely to have like loss of function mutation without any cause, you find a mutation there that probably it's the causal mutation. And also with this gene expression stuff, in the early days we didn't find any equitals for genes that are basically crucial for the maintenance of a cell as such, regardless of the function of the cell, but the maintenance, because those things just cannot tolerate any variation, because then the basic biology will just fall apart. But there were like two other questions, three.
Yeah.
Okay.
I'll just answer. Is the red line on that graph an observation?
Yes.
So is that like super sharp, like upward trend and then the flattening off? Is that just how good state screening? Like you just capture this whole
Maybe.
itself.
Maybe it can be, yeah. Yeah, but it's also like you have to keep in mind that it's the sample size. While we are working with 50,000, it's only 30,000 females, and of them only like not many develop breast cancer. Then you're certified by the polygenic risk score, so you end up with like maybe 100 or 200 in each of your categories. So that's why you also get a lot of fluctuation because the sample size isn't particularly large. Well, I think this is striking, and if you show this to policymakers, they immediately want to implement polygenic risk scores in healthcare. The last one.
Again, that breast cancer seems like a good problem. How would the kind of farming with cumulative incidence look instead for family history? And then if you had sort of less PGS categories and a few more family history categories and they were combined, how might?
So I would say the family history is important if you are carrying not a high load of polygenic score, but you have this high impact mutations. Like for example, in breast cancer, it's this BRCA1 and 2. Probably everybody's known this or heard this Angelina Jolie story where she had this mutation, and that's why he had like 80% risk to develop breast cancer by age of 50. And there, of course, the family history makes a complete sense. But for example, here with polygenic risk scores, because even though the parents may have a normal distribution, but if you are super unlucky, you may have like this bloody, high end of the distribution, regardless what's the family history. So in this case, the family history doesn't really help you a lot, because with the polygenic risk, there is just it's not that there isn't even any family history available. And also in the modern world, in Facebook, we are sharing information about, where we went on vacation or whatever, got a new cat or something. But no one is actually sharing this very sensitive information of getting a disease. And it's even more complicated if we have in this, I don't know what's the English word, where there is like this complex family structures that is like my children, and then it's your children, and then it's our children, and then somewhere else children living in one place. So actually even, you don't even maybe sometimes know whose genes you carry and to make the kind of whose family history you have to follow. So that's why it's the same with any of those, for example, the education and parental education or socioeconomic status, it's information that is not just available to use, but the genetics is kind of very uniform stuff that you can use.